Hello everyone. Today we will learn about a very interesting online server which is known as Hop. When you will search it on the Google, you will be able to see the option of Welcome to Hop. Click on it and it will take you to the home page of this server. Hop is an easy-to-use web service that analyzes the structural effects of a point mutation in a protein sequence. Input your protein sequence and the mutation, and Hop will collect and combine available information from a series of web services and databases and will produce a report complete with results, figures, and animations. Now you will click on the Try Hop and it will take you to a window where you are supposed to paste the sequence of your protein. Now you will paste here the sequence of the amino acids which is belonging to the normal protein. In any of the case, you are never going to paste the sequence of mutated protein because whenever you have to analyze any specific SNP, then this server will give us option to select that particular position of SNP in the normal protein, and then we will tell it that at this position the this mutated amino acid is present. So for any case, for any type of single nucleotide polymorphism, you are supposed to paste here the sequence of your normal protein, and you can see below this window the multiple options like select a residue to mutate, select mutation, and select modeling method. After pasting your protein sequence, you will click on the next. It will show you the whole sequence of your protein and the number of amino acids as well. Now suppose you are having a mutation in your case which is localized at position 40 and there is amino acid which is A, so you will select that position, that particular position. In my case I selected, suppose we select the position 23 where R amino acid is present, so you will put the cursor on at this position and will click it in order to select it. So, and you selected, and here it is telling you that you selected the arginine at position 23. Then you will click on next, and then it will show you the names of all the amino acids which may be present at that particular position which you have selected in your protein sequence. Suppose the amino acid arginine has been mutated to tryptophan, so you will select the tryptophan, or if it is changed to threonine, then you will select the threonine, and so on, and then click on next, and this will give you the option of the submit, and you will click on submit. The server may take 1 hour in predicting the results of your query, and definitely I cannot show the whole result page as a whole in my PPT, so I separated it in parts, and the results are very interesting. Initially it gives us the comparison of the structure of the wild type amino acid which was present initially in the protein before the mutation, as well as the amino acid in which the wild type amino acid has been changed or mutated as a result of a particular single nucleotide polymorphism. So this is clear that the structure of the wild type amino acid and the mutated amino acids is very much different. In wild type amino acid there is only single ring, while in a mutated amino acid it is double ringed, sort of double ring structure. Like the structure of mutated amino acid is different, is much bigger than the wild type, and the characteristics of these two amino acids are also shown below the figure. Like you can see here that each amino acid has its own specific size, charge, and hydrophobicity value. The original wild type residue and newly introduced mutant residue often differ in these properties, and the mutant residue is bigger than the wild type residue, and the wild type residue is more hydrophobic than the mutant residue. So, and also if suppose If this amino acid is present in such a domain which is interacting with the other domains of the protein, then definitely if this amino acid is changing with the amino acid which has low hydrophobicity, then definitely the hydrophobic interactions between the domains maybe might alter as a result of this SNP. And here are the three possible domains which are predicted by Hope server in which that particular mutation may be found. Like the first is enoyl coenzyme A hydratase or isomerase, and second is fatty oxidation complex alpha subunit FADB, and third one is CLPP or crotonase like domain superfamily. Now the mutated residue is located in a domain that is important for the activity of the protein and is in contact with the residues in another domain. So it is possible that this interaction is important for the correct function of the protein, and the mutation can affect this interaction and as such affect the protein function. Now the amino acid properties are given here. The wild type and mutant amino acids differ in size, as we have already seen with their with reference to their structures. And the mutant residue is bigger than the wild type residue. The wild type residue was buried in the core of the protein. The mutant residue is bigger and probably will not fit into the core of the protein. The hydrophobicity of the wild type and mutant residue differs, and the mutation will cause loss of hydrophobic interactions in the core of the protein. Now this is the ribbon representation of the three-dimensional configuration of your wild type protein. First figure is very colorful, and then in which the alpha helix is shown with blue, beta strand is shown with red, and turns are shown in green. But the picture which is given below is in gray color, and only a specific part is colored in magenta. This is actually the point where your particular SNP is changing, is creating a change. Here the particular SNP, the mutation has occurred, and this is the closer view of your SNP of your mutation in the three-dimensional structure of the protein. The rest of the protein is shown in gray color, while the green part is showing the wild type amino acids which are localized already, which are present at that particular site of mutation around the mutated amino acid, and the red part is actually showing the mutated part. And you can find you can find the various views of this particular mutated point through this server, and you may retrieve these structures, and you may represent them in the form of a figure, and in which you can represent the structures obtained through multiple mutations. You can represent all the structures in one figure, so that so it will make it easy for the reader to compare the differences in the mutated proteins. Thank you so much.